Great. Well, welcome everyone. I'm Corey Garza, and you're now attending Taylor Eddy's thesis defense here at Moss Landing Marine Labs. Just a few ground rules for Zoom before we get started today. So, just so you know, all audience participants are going to be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. And then there's going to be an opportunity to ask questions live at the end if you're on Zoom, just like a normal seminar. Once the speaker has finished their talk, you can use Zoom's raise hand feature to notify the meeting host that you'd like to ask the speaker a question. And so the raise hand feature is located under the participants tab at the top of the Zoom window. Okay. Sense why calling it a journey of first as we go along here today. So I've known Taylor about 8 years now at this point. I can't believe it's been that long already. And just so you know, I first met Taylor when she was an REU student in our very first REU cohort here at CSU Monterey Bay. And interesting enough, she actually did her REU research at Moss Landing Marine Labs with Jim Harvey back when he was running the vertebrate ecology labs just a few years prior to him moving into the directorship there. You can see her with their very first cohort, and they're presenting some of her work on marine mammal. I think she's working with humpback whales back then on there. She kind of went on for a couple of years there, finished up her bachelor's, went off to do a little bit of work for a bit, and then approached me a few years back about coming to graduate school. And so we had just received an award for one of Noaa's cooperative science centers, the Center for Coastal Marine Ecosystems here at Monterey Bay. And so Taylor was part of our first ever Noaa CCME graduate student cohort. And so she was part of an initial group of 3 graduate students and 3 undergraduates who we took in that year. So you're actually seeing them at our very first training symposium with the rest of the center members there at the Gulf Coast Research Lab in Mississippi. And then you can see her there later on with one of our second year CCM undergrads, Riley Young, on the back of the boat there with me. And so Taylor spent the last few years conducting her thesis research at Catalina Island. She got quite a bit out of the Noaa program. It was a really fun program. I think she'll admit she got to do quite a bit beyond science. She got to take a look at how policymakers actually get to engage with the type of work that we do. And so you're actually seeing her there on the left at the Capitol Hill Oceans Week, which she got to go through via support from Noaa CCME as part of her participation with other students from around our center. And then she's there presenting to our associate director at the CCME meeting. Now, Taylor actually came into a unique time in my lab where, as some of you who know me know, I do a lot of marine landscape work with digital photography, total stations and GPS units. And at this point when Taylor started, we had just started our first drone program. And so Taylor was actually instrumental in helping us get our drone program up and running here at CSUMB. In fact, I believe she was our first student pilot at CSUMB to have an FAA certification. And so she incorporated a number of drones that we have on our campus here into her thesis work. And so she'll be talking more about what she did there, but she really kind of set up the sort of the launching pad for our drone program and sort of what it is today. And, just like, I like to do with teaching, Taylor's also become a teacher herself. She's also passing on her experience to first-time researchers. And so she's been really sort of generous with her time and sort of passing along all that she's learned her time, both as an REU student and a Noaa student, to students who are coming after her. So you're seeing there on the left, they're working with one another REU students, Madison Hurd, when Madison was doing some work at Mbari. And they're on the right there. You're seeing her with one of our newest Noaa students, Jason Gronich, kind of introducing him to drones, like just how to get him out of the box in this case. He's actually now a fully licensed FAA drone pilot, and so he'll be sort of coming into our lab to take over some of our drone work pretty soon. So Taylor's actually had quite a bit of experience here. I think she's done quite a bit here, and she's going on to bigger and better things with the USGS now as a full-time employee. So I just want to say before we get started today, good luck, Taylor. Best of luck in everything that you're doing with USGS, and I'm sure you're going to do a great job today. And so with that, I'm going to turn it over to Taylor so she can begin her presentation.
Thank you so much, Corey. That was great. see if I can you stop sharing your screen so I can get mine? Okay. Thank you very much for coming, everyone. So today we're going to be talking about the multiscale habitat, multiscale habitat use and effect of resource availability on California spiny lobster, Panulirus interruptus population success. So the habitat and resources that are available where a species lives has an effect on many aspects of their life, such as growth, survival, and reproduction. And a lot of that is fueled on what's available in the habitat, so the food resources that they have. But sometimes one habitat is not enough to support the population without outside resources, so they have to make a move. This can be on a large scale, like the salmon above here, which spawn in rivers, but then they move offshore to feed, and they repeat this seasonally. So that movement offshore, they get introduced to some new food resources. There's also a smaller scale example here with the Pisaster, the sea star that's on the bottom. This sea star moves within the intertidal as the tide moves. So when the tide is low, the sea stars are in that low intertidal zone. But as the tide begins to rise, the sea stars also rise up into the higher intertidal, and they forage on the species that are found in that upper intertidal while the tide is high. And as the tide retreats, the sea stars follow back down to avoid being exposed to the air. So they follow this daily movement of the tides. And this movement, both in the salmon and the sea stars, it subsidizes their diet by introducing some new species for them to forage on. And so they have these new food resources. So the salmon moving offshore and the sea stars moving up is an example of predators migrating to the prey. But also, when these salmon come back up the stream each year, they have a new predator that's waiting for them, the brown bear. These salmon then become a subsidy for the brown bear diets as they make this migration. In either case, this diet subsidy leads to an increase in predator population due to this prey influx. Another species that makes these movements into other systems is the California spiny lobster. The California spiny lobster is often found in kelp forests from Mexico to Southern California. And in these kelp forests, they're actually a keystone predator. So they control the urchin population. When they're present in high numbers, they forage on these urchin populations, and that changes the habitat that they're in. It allows for more kelp to grow because there's the kelp isn't being foraged on by the urchins. Not only are they this ecologically important keystone predator, they're also economically important. They're the third highest grossing fishery in California, and there's also a large-scale recreational fishery. But what people sometimes don't realize is that lobsters actually move up into the intertidal zone in their summertime during their reproductive season. So this is from a study done in the 1980s by Carlos Robles in Southern California. And he set up these underwater cameras in the intertidal after he saw the spiny lobsters in the intertidal at the nighttime high tides. So we wanted to see what was going on. So in A, we have the tide rising. There's a lot of water movement and no lobsters yet. And in B, as that tide continues to rise, we start seeing a few lobsters moving up into that intertidal zone. And C is the slack tide, which is the highest point of the tide where there's the least amount of water movement. And this is actually when Carlos Robles found that there's the highest density of lobsters in the intertidal. And finally, in D, as the tide retreats, the lobsters also retreat back down to the subtidal. So when he was making these observations, he found that the lobsters were foraging on mussel beds. But those mussel beds here were pretty sparse, but they were preferentially foraging on them. So we wanted to see what kind of effect these predators were having on the mussel population. So we set up these cage experiments where one part of the intertidal was caged completely, blocking out any predator access. And there's also a cage control, which still allowed for predators to forage within it, including the spiny lobster. What he found was that when he removed the cage at these sites, there was a patch of mussels that were able to grow under the cage where there was no predation, but there is no mussel growth in the rest of the intertidal. So in the absence of predators, the lobsters, the benthic habitat changed, which means they're now also in a keystone predator in the intertidal, controlling this mussel population. And without the lobsters there's no persistent mussel, or there's less mussels in the intertidal, and it's more an algal turf dominated system. So this was actually part of a larger study down on Catalina Island. As Corey mentioned, this is where my research took place. So Catalina Island is off of Los Angeles, where that green dot is on the map. And specifically, this study took place at Big Fisherman's Cove and Bird Rock. There are these sites here on the map, indicated by the red dots for Bird Rock and the blue dots for Big Fisherman's Cove. And these permanent study sites, there's 3 in each area, and they were set up in the early 1980s. And each of these dots represents one 20-meter long transect by 1 meter wide. And Carlos Robles did these studies in the early 80s, but then something unique happened, and USC Wrigley Marine Protected Area was designated in 1988. So this site, Big Fisherman's Cove, became this protected area. And this is actually a really unique data set for protected area assessment, or MPA assessment, called the BACI. It's the before after controlled impact paired series. What that means is we have data from before an MPA was established and after an MPA was established at a control site, which is Bird Rock, which has no change, and Big Fisherman's Cove, where an MPA was placed. And this allows for researchers to see what kind of effect the MPA was having on the species of lobsters. So in 2013, the Marine Landscape Ecology Lab was awarded a grant to go study how this MPA was performing for lobsters. They were shocked to find that the overall condition of lobsters was better outside of the MPA, out at Bird Rock, than inside of the MPA. And they thought that was interesting because we think that the MPA would be protecting the species, but they found there's more lobsters, more females outside of the protected area, and more of the females that they found were reproductive. So they thought this might be due to differences in the habitat and the resources that were available. So to get at that, one of the past grad students, Sean Wendell, did a habitat assessment at Bird Rock and Fisherman's Cove and found that out at Bird Rock, there is more of this preferred prey item, the Mytilus californianus, that mussel. And inside of that protected area, there were no mussel beds for the lobsters to forage on. So from that, they concluded that it's possible that the habitat might have something to do with it, but they wanted to take it a step further and look at the diet of the lobsters. So another grad student, Mary McCormick, did a diet study that further confirmed that when the mussels are present, they're the preferred prey item. And this likely accounted for the difference in demographics of the lobsters. So here we have the graph at Big Fisherman's Cove. the lobsters are predominantly feeding on crabs, and at Bird Rock, they're predominantly feeding on mussels that comprised about 75% of their diet when looking at the intertidal prey species. But what we still don't know is if this is a persistent trend. So this was just to look at one snapshot of one summer diet that included that important intertidal resource. But we wanted to look at what the diet and what these changes were happening, what the difference were seasonal and annually. And we also wanted to look at the diets of how the diets of the lobsters varied between sexes seasonally. So that brings us to the goals of my thesis research. And the first was to assess the population of spiny lobsters at both sites and assess the differences in habitat between the sites, to assess the seasonal diet of lobsters between sites and sexes in Catalina, and finally combine all of these results to evaluate the reproductive success in relation to resource availability. So for the remainder of this talk, I'm going to break it up into 3 different sections, and those sections will be indicated by these symbols that are above the boxes, and those symbols will be in the right-hand corner. So the first is the lobster population metrics, where we're going to look at how many lobsters and what the condition of the population is. Then we'll go into the habitat characterization, and finally the diet assessment. So starting with those lobster population metrics. In the summer of 2017 through 2019, and also the winter of 2019, we went back to those sites, those 20-meter transects, and we did observational transect swims and lobster collections. So we went diving, and on our first pass over those benches, we had the intertidal and the subtidal that were both 20 meters. And so the first pass, we counted the lobsters that were in that intertidal zone. And then the second pass, we would collect all the lobsters that we could. And we did the same for the subtidal and did that for all 3 transects at both sites. When we collected the lobsters, we brought them back to the boat, and we recorded the location that they were found. We measured the size of the lobsters, recorded the sex, the reproductive stage of the females, and we also removed a leg, which is a non-lethal way to sample, and they do grow the leg back, and we were able to get a muscle sample from the lobster legs. So this is an example of a lobster that we found at Big Fisherman's Cove. This is a female that's gravid that has those eggs down in the tail, that orange egg mass. So for the statistical analysis, I used an ANOVA to assess the population density between the sites at Bird Rock and Fisherman's Cove, and also between the intertidal and the subtidal at these sites. I also used a chi-squared test for homogeneity to assess the differences in sex ratio and reproductive condition between the sites. So this is the summer data from 2017 to 2019. And in all these following graphs, we're going to have Big Fisherman's Cove on the left and Bird Rock on the right. In this case, the intertidal is represented with a white bar, and the subtidal is represented in the gray bar. And this is the density of lobsters found on these transects. And what we found was that there's a higher density of lobsters at Bird Rock than at Fisherman's Cove, but at both sites, there was a higher density of lobsters in the intertidal than the subtidal. And this is the results from the winter, summer and winter, but it's just, we're looking at the subtidal because the lobsters don't have access to the intertidal during the winter because of winter storms and the tidal heights. So we have at Big Fisherman's Cove, the density of lobsters in the subtidal was the same between summer and winter, but at Bird Rock, the density of lobsters was higher in the winter than in the summer. And this is likely due to lobsters at Big Fisherman's Cove moving out of our study site in the winter time, where at Bird Rock, those lobsters that were in the intertidal, instead of moving out of our study site, they moved into that subtidal site where we were able to count them. So now this is the summer data from 2017 to 2019, and this is from the lobster collections. And we were able to collect 118 individuals in this time period. 84 of those were at Bird Rock and 34 were at Big Fisherman's Cove. So here the white bars indicate the female population and the female lobsters that we caught, and the gray bars are the males. So here we see that in the intertidal at both sites, there's a higher proportion of females than males, but there's also overall a higher proportion of females at Bird Rock than in Fisherman's Cove. And of those females that we collected, we found that there were more females in the more reproductive females in the intertidal than the subtidal at both sites. Those reproductive females are represented with those white bars, and the non-reproductive females are represented with the gray. So at Bird Rock, at Bird Rock, there were more reproductive females than the entirety of Big Fisherman's Cove. So now that we've found that there were more lobsters at Bird Rock, more females and more reproductive females, we wanted to see if those habitat differences also remained the same. So to do that, we used methods based on Robles et al. 2010, and we created these photographic transects where we took pictures in the intertidal, and we used georeferencing equipment so that we can stitch them together using a program called Arcgis. And this allows us to spatially reference each of these photographs and create these photographic transects that you can see up in the right-hand corner. So this is the methods we used in 2017 and 2018, but in 2019, we implemented the use of drones. So for this, we were looking at those same intertidal transects, and the software that we were using didn't allow for autonomous flight below 10 meters, which means that I manually flew the drone because we wanted to get a height that was comparable to where we were holding the camera. So I flew the drone between 2 and 3 meters and followed a path that's in this upper left-hand picture in green. We have where I flew the drone away from me for 20 meters and then brought it down lower into the intertidal and brought it back, taking photos overlapping using an on-screen grid. And after we took all of these images, we ended up with something that looked like this, where we have this green line that's the path that the drone flew, and each of these blue dots is a photo taken manually. All of those photos were then uploaded into Pix4D, and in this software, it uses structure from motion technology to stitch together all of the photographs and can render these flight paths in 2 and 3 dimensions. So in the bottom left, we have a 3D rendering of one of the transects out at Bird Rock, where the green on the left is actually the low intertidal zone. And as we move to the right, we're moving up higher into that intertidal. So each of these missions, flying the drone, took less than 10 minutes to collect all of the data, where when we were using the photographic transects and georeferencing each of the pictures, each transect took about 20 minutes. So we're cutting the time it takes to collect all of the data in half. So this allowed us to, instead of taking 2 full low tide cycles in the morning to collect all the data, we now can collect it all in one morning with less equipment and flying drones in the intertidal. So from the Pix4D, this is now an image in 2 dimensions, and we take the photographic transects and those Pix4D images, and we bring them into ArcGIS for analysis. So I use supervised image classification in Arcgis, which takes advantage of the fact that in these photos, different species reflect light at different wavelengths, meaning they show up in different colors. And I was able to train the software to understand that pink pixels that were coralline algae showed up as pink pixels. And I was able to tell that to this program, as well as black pixels being these mussels, red pixels being red algae, and so on for all of the species that occurred in these transects. And once we were comfortable with these training runs, we used maximum likelihood classification, which then took those training samples and with those with the wavelengths associated with each species and applied that to the entire photograph. And it analyzed each individual pixel and classified it as a species. We were then able to extract percent cover by selecting just the pixels that contained a species. For example, coralline algae. If coralline algae encompassed 100 pixels of the 1,000 pixel image, then that would be 10% of the cover. So we used this analysis to get percent cover of each of these images. We then used this percent cover and I performed a permanova to test if there were distinct differences in the benthic habitat composition between Bird Rock and Fisherman's Cove, and NMDS, which analyzes the relative proportion of communities within each site. So that data is represented here. On the left, we have the graph of the percent cover, and I took the averages over the study period. And we found that Bird Rock is predominantly a turf algal site, and Big Fisherman's Cove is pretty evenly spread out between Thalamus, which is a barnacle species, turf algae, and some other algae that are on the right-hand side. But a striking difference was the Middleus, the mussel population there. So Big Fisherman's Cove had less than 0.5% of mussel cover, and Bird Rock had about 2% mussel cover for the entire area, or for all three transects. But there was only one mussel bed in our study site, and if we just looked at that one transect, it ended up being 5% cover at that transect. On the right here, we have the NMDS plot, and to interpret this graph, the farther away these points are, so each of these is one of the transects studied, the farther away they are, the more different they are. And so these results show that they are distinctly different habitat types. This is likely due to the high presence of turf algae at Bird Rock and also that presence of mussel. So now we wanted to see how the diet changed annually and get the seasonal and seasonally. So in those same years, 2017 to 2019, and in the winter of 2019, we used the mussel from the leg that we collected. And we also collected prey items from the intertidal, which were mussels, crabs, and limpets. And in the subtidal, we collected urchins and snails because we know that the lobsters forage on these items. We then processed these samples and we dissected out pure mussel tissue from each of these species, and we dried it and sent it off to a stable isotope facility at UC Davis and Idaho State University for stable isotope analysis. So what are stable isotopes? These are naturally occurring isotopes of elements, and in this case, we used nitrogen and carbon. And this uses the ratio of nitrogen 15 to nitrogen 14, that heavier isotope to lighter isotope. And the same with carbon 13 and 12. And the ratio of these isotopes change in a predictable way. And this allows us to assess diet over a longer period of time when we use these muscle samples compared to traditional methods such as gut content analysis, which that just gives us a snapshot in time of what the lobster would have in its stomach at the time that it was captured. So what can we do with these? There's a program in R called Stable Isotope Bayesian Ellipses in R, short is CYBER. And this program fits an ellipse around a group of data using Bayesian statistics. And we can use these ellipses to estimate the niche space and compare between groups that we want to look at. So we can compare this isotope niche space. So on the left here, we have these ellipses that are created in CYBER based on the data, the isotope data of based on the isotope data. So the larger the niche space is, it generally means there's more prey items incorporated into this niche. The smaller it is, the less prey items there are. And we're going to be looking at two different kinds of graphs that will both depict this isotope niche space. On the left are the ellipses, but on the right are the plots of the area of this niche space. So that each of these ellipses corresponds to a dot on this graph. And the gray boxes around it indicate the error associated with these points. And another analysis we'll be using is stable isotope mixing models, which works off the premise of you are what you eat, literally. So if you know the prey's isotope signature and you know the predator's isotope signature, we can estimate the proportion of diet that each of those prey items play in the predator's diet. This is kind of like mixing paint. So if you have purple paint, blue paint, and red paint, we can analyze the purple paint to see the proportion of blue and red that makes that specific purple. This is the graph that we'll be using today with the mixing models. And the further we move on this x-axis, the higher a role the prey plays in the lobster's diet. So in this case, crabs comprised about 80% of the diet, and mussels comprised less than 25%. This is an example from that 2013 study. We'll be looking at the data collected in my study. So of the prey that we collected the same species at Fisherman's Cove and Bird Rock. And so this is the potential prey niche. So this is all the species that we collected at Bird Rock and Fisherman's Cove. And the prey niche here is pretty equal. And if all else were the same, we would expect that the lobster's niche space would be equal at each site. But that's not what we found. So here we have the ellipses at Bird Rock and Fisherman's Cove. Each individual collected at Big Fisherman's Cove is represented by a triangle, and each individual collected at Bird Rock is represented by a circle. Those collected in the winter are in red, and those collected in summer are in black. And what we see here is that there's this expansion in the summertime to a larger niche area where there's this increase in nitrogen and decrease in the carbon isotopes. And so both sites, they're moving the same way, which indicates that there's a new prey resource being added in the summer that isn't available during the winter. So these are the ellipse areas of the ellipses, or the areas of the ellipses that we were just looking at. And the ellipses in the summer are larger than in the winter at both sites. But at Big Fisherman's Cove, the ellipse is even larger than that in the summer at Bird Rock. This is likely due to the lobsters at Big Fisherman's Cove being more generalist feeders. So anything they come upon, they're eating, where at Bird Rock, they might be specializing on one or on a smaller subset of prey items. The winter niche of the lobsters at both sites are actually relatively equal, which indicates that they're feeding on the same prey items. But there's less prey diversity available in the winter than in the summer because those niche areas are smaller. I then analyzed the differences in the annual niche size of the lobsters. So the 3 bars on the left are from Bird Rock, and the 3 bars on the right are from Big Fisherman's Cove. And we see that the niche space each year is smaller at Bird Rock than at Big Fisherman's Cove, indicating that they're foraging on less of a variety of species, or likely preferentially feeding on a certain species or set of species. So then I looked at the mixing model results. So these are the results from the summer at Bird Rock. We have, for both males and females, the lobsters were foraging mainly on the subtidal prey items, those snails and mussels. And each of these peaks is represented by a picture depicting what they are. So the highest proportion of diet for both males and females were these urchins. The next highest was the snails, which is not surprising because they have more access to the subtidal than the intertidal. But then we see with females, there's this 7% of their diet they're foraging on mussels, where only 4% of the diet of males are comprised of mussels. So they have this subsidy where they're foraging in the intertidal, feeding on these mussel beds, which subsidize their subtidal diets. But there's this separation between males and females, where the females are preferentially feeding on the mussels more than the males are during the summer. At Big Fisherman's Cove, we have this same plot, but we have most of the diet of both males and females are urchins, which comprised about 75% of the diet, and the snails, which comprised about 20% of the diet. And those diets of the males and females are very similar. So there isn't that separation between the sexes. For the winter diets, we excluded the subtidal prey items because they don't have access to this. So we were only looking at the snails and urchins as their subtidal prey. So here, the males and females at Bird Rock have very similar diets in the summer or in the winter, when they had those very different diets in the summer, but now they're foraging on the same proportion of snails and urchins. And at Big Fisherman's Cove, the males and females were both feeding on about 50% urchin and 50% snails for their diets. So they also had very similar diets when you compare them, which is the same trend that we saw in the summer where they had similar diets. All right, that was a lot of information, and now we're going to wrap it up. So we found that in the summer, spiny lobsters moved from the subtidal to that intertidal zone. This intertidal zone subsidized their diets during their reproductive time, the high energy time of reproduction. In the winter, the lobsters were foraging on similar prey resources between the sites. And because of wave exposure and the proximity to longshore currents, Bird Rock and Fisherman's Cove have distinct habitat types, where there's mussels present at Bird Rock, but not at Big Fisherman's Cove. And these mussels are having a disproportionate effect on the reproductive success of the lobsters. Even though it's a small proportion of the diet, it was 7% of the diet of the female lobsters. they're having a large effect on the reproductive success. So at Bird Rock, we're seeing this increased population density and the increase in reproductive females. And this is likely due to that diet subsidy. And lobsters are opportunistic predators. The absence of mussels at Fisherman's Cove decreases the value of that intertidal subsidy. But it's actually likely that mussels played a larger role in the past than they do today. This is actually an image from Bird Rock in 1984, where we see these large expanses of mussel beds that we don't see today. So this is Bird Rock in 2016. There's been a substantial decline in the mussel bed presence. So this subsidy is disappearing. But this isn't unique to just Catalina. On the mainland of Southern California, this is Crescent Bay in Orange County. And in 1973, there were these huge mussel beds with layers upon layers, and the sea stars were feeding on them. But in initial analysis using historical panorama and automated classification, they found that these mussel beds are retreating. So the mussel beds in each of these photographs are represented in red using that color classification in these images using Arcgis. So from 1972 to the present day, we see this upward compression of the mussel beds. So all throughout California, this subsidy is disappearing, and we don't know the effect that this will have on the lobster population. So with these mussel beds declining, the value of the intertidal subsidy is decreasing. And when we're creating protected areas, it's really important to consider the seasonal and seasonal variation in spatial foraging. So in the summertime, they're foraging on these really small scale features like the mussel beds, where in the winter, they're foraging on these larger scale urchins and snails that are found throughout the kelp forest. And while we saw the unexpected for lobsters, other species within this MPA are actually benefiting from this protection. So species like flatfish and leopard sharks are doing really well. But this study provides an instructive example for understanding why MPAs might not work for some species, but they do for others. So it's really important to begin considering the nutritional value of the MPA in addition to looking at the population and the numbers that we see within the number of species we see within the MPA. And with that, I'd like to thank the Noaa Center for Coastal and Marine Ecosystems, who is a huge funder of this project that funded my research. And without them, I wouldn't have had the same experience. As Corey said, I got to do a lot of really incredible things, and I learned a lot by participating in this program. Coast was also very helpful in matching me with some incredible undergrads that I got to work with. USC Wrigley Institute for Environmental Studies, that was the house that I had in Catalina. We worked from this research station, and they were so accommodating, and it was really incredible to be part of their research group. And I want to thank Dr. Corey Garza, my advisor. For 8 years, I've been working with you, and you've been really helping me to grow to be a better scientist. John Geller, thank you for your insight and your help through navigating all of the Moss Landing, all of the research, and all of the input that you've given throughout this whole process. Scott Hamilton, also a big thank you for all of the input that you've had throughout this whole process, as well as Steve Litvin, who I've also known for a very long time since undergrad, who's really helped me develop and become join these programs. It was actually Steve Litvin who encouraged me to apply for that REU that first year. I also want to thank the undergrad researchers, Riley, Rose, and Jason. You guys were incredible and I couldn't have done it without you. CSUMB staff, Moss Landing staff, USC Wrigley staff. There's so many people to thank and none of this would have been possible without them. And the stable isotope facilities, UC Davis and Idaho State University for running my samples, sometimes at the drop of a hat when I had a conference deadline, and all of the volunteers who truly did make this possible. And you guys are all incredible. I know at least some of you are watching today, and I really appreciate all of the help and sacrifices that you made for helping me collect all of the lobsters and intertidal species. And finally, the CSUMB MLEL lab for listening to all of my ideas and helping me to create this presentation today. And all of my family and friends, I told myself I wasn't gonna cry. All of the memories that I've made over the last 4 years at Moss Landing and all of the support that I've gotten from all of my family. You guys are all incredible. And thank you very much for everything. And with that, I'll take any questions.
Great, well, thanks, Sarah. If we can, maybe do a little virtual hand clap on here before we take questions. We can use your little thing there. But great, Taylor, thanks so much for your talk. And I think with that, we are ready to take questions here. So let's go ahead and get going. And I'll go as I see them in order here on my list. So first up, it looks like Patty and Petro.
Great job, Taylor. A lot of work and very impressive. I wonder, in light of both the sort of micro scale and then the some of the larger scale obvious differences between your two sites, Bird Rock and Fisherman Cove, what do you think that does to the idea of using them as a bocce model, sort of for testing MPA effectiveness, given the differences you saw in the sites themselves?
Yeah, that's a really good question. So one of the advantages of this model is that we have data from before the MPA was established, and we have years of those data. So we can see the sort of trends that were happening before the MPA was put in place at both of these sites. And we can look at the trends before and after the MPA. So that helps us to tease apart those environmental differences between the sites.
I see, but if the test is whether there's an MPA or not, and the sites are different to begin with, then how do you tease apart whether the differences are due to the difference in the sites or the fact that there's an MPA? I guess was my question.
Well, we can look at the trends at both sites, so we have that control. So we can look at the trends at both of those sites, and yeah, they are going to be different and they're going to change at some different rates, but we can look at those rates over time and see what those changes are, and it's just one way to tease out some of the environmental.
I see. Great job.
Thanks.
It looks like we have Emily Pierce next.
Hi, Taylor. Great job. Two quick questions. Now that I live in Maine, I wanted to ask which lobster is better, the lobster that you guys have there or the lobster that we have in Maine? And what was the coolest organism you found while diving other than lobsters?
Definitely the California Spiny Lobster. They have a very special place in my heart, and I spent a lot of time underwater with them, and they're gonna win every time. I've also heard that they taste better than Maine lobsters, so I haven't tested that one yet, but maybe that'll be a future. One of the coolest things I saw was an abalone the size of a dinner plate, and I think that you would get a kick out of that. But at Bird Rock, that the abalone that we find out there are huge, and it was really incredible to see that, especially with the state of the abalone that we have through all of California.
Awesome, thank you. Come out to New England and we'll get you some
Hi,
Maine
Taylor.
lobster. Steven says hi too.
Hey, looks like Scott Hamilton's got his hands up.
Hey Taylor, that was great. It was nice to see it all integrated and come together really nicely. I was starting to think about the kind of the tissue turnover time in the lobsters for your isotope results, and I couldn't remember exactly the timing of when you collected the lobsters in the, I guess, the reproductive season, right? Because if you collected them really near the beginning of the season, then they wouldn't have had many opportunities to eat mussels and kind of incorporate that into their tissues. But if you collect them at the end of the season, then they might have had longer. So I was kind of curious how the timing of your collections might have affected your interpretation of the importance of the mussels.
Yeah, based on those turnover times, we actually go out at the end of their reproductive season. So their reproductive season generally starts at the end of May and goes through about July, early August. And we would go out at the end of July and collect the lobsters for the mussel samples.
Okay, so that's so this estimate's then kind of potentially near the maximal sort of percentage of that.
We give them the time to eat their the forage in the intertidal, and we can see that prey come up in their diets.
And do we know how long in general, sort of what that turnover time is for those tissues? Is it a month or 6 months, or sort of how far back is that integrating information about what they've been eating?
About 2 to 3 months.
Yeah. Awesome, thanks.
Of course.
Hey, any other questions out there? I don't know, Scott, do you still have your hand up?
Well, I'll ask another one if no one's got. I was trying to put it down, but I couldn't
Okay.
figure it out. But hey, I'll ask another one since no one else is asking questions. I guess my other question was, has anyone done any complementary like gut content analyses of the lobsters? I was just curious, like what other prey items they may be eating commonly that you weren't able to sample, or if there were any.
There have been gut content analysis studies. Corey, another student, did one a while back, correct me if I'm wrong, and found that the major prey resources were in the summer. We found the shells of mussels, limpets, crabs, a lot of the invertebrates. Some studies from a while back suggested that potentially barnacles were a prey resource, but in the gut content analysis that was done more in Catalina at these sites, there was no evidence of barnacle foraging. But I think based on the gut content analysis studies done, we got most, if not all, of the prey resources that the lobsters were foraging on in the summer and winter.
Looks like Pat has a question.
Yeah, I won't turn my camera on for this one. Given that the mussels seem to make up such a small portion of the total diet, and the fact that you are surmising that that contribution of the mussels as a prey item is so important to reproductive success, what do you think it is about eating mussels that helps the lobsters be more successful reproductively? Is it just a matter of more food or something special about that food item?
I think it's actually the quality of the food item. So mussels have a really high protein content. They also have a lipid content in them that some of the prey items, just the subtitle prey items, wouldn't have. So it could be acting as this sort of catalyst for some internal successful reproductive season. So without it, the lobsters can't start this, they can't start this process, or it has some maybe type of lipid that other prey items wouldn't have, which is why I think a future study would be really interesting, would be to look at the composition of mussels and the nutritional value that they do provide, and try to parse out what exactly it is that's having these lobsters, helping them to be more successful during their reproductive season.
Looks like Travis Leggett.
Hi, Taylor.
Hi.
This is very exciting to see you give this presentation today. So my question is rather open-ended, and so I hope you're still able to give a satisfying answer. But so we're starting to see spiny lobsters pop up very infrequently along the Central California coast. And I was wondering if you had given any thought to how their reproductive success based on their diet may change, considering the different food that may be available up on the Central Coast.
Yeah, it's very open-ended, and the possibilities are really endless. It could be that there's some subtitle prey items that are introduced to this species that actually also promote this increase in reproductive condition. It could be that there are more mussel beds in Monterey than in Southern California, so they would be foraging on these same mussel beds. So we could actually see an increase in reproduction if the physiological constraints are continued to be met. So if the ocean does stay warm enough up here for the lobsters to persist. But I think there's still a lot of unknowns, and really I think anything can happen, but it could be that they do very well because there are more of those larger mussel beds in Monterey than in Southern California.
Right on. Yeah, cool. Very satisfying answer. Thank you.
Thanks.
And congrats and good job.
Thanks, Travis.
Great. Well, I don't see any other questions, and it's 5 o'clock. So I think we might let Taylor off the hook here, because I believe she still needs to speak with her committee. So that maybe we can give her a round of applause for her talk today. Great, Taylor. Yeah, you might want to stop sharing your screen.
There we go.
Great. Well, I think
Thanks
at
all
this
for
point,
coming.
yeah, thanks everyone for coming. So I believe at this point, you talk to your committee, and then it'll be time to chat with other folks.